was. The other is to show you the useful information and also the limitations compared to using other genetic models like pea plants and Drosophila and such. And the second half of it is really to sort of talk about genetic counseling and what you can do now. And we can also think a little bit and talk a little bit about how this is going to really be changing, I think, in the near term over the next several years. So we started out, we're looking at some of these. And so let's just kind of go back and look at this. And now what I expect you to do on an exam is I'll give you this pedigree, not with all the information on it, and just ask you, give you this one or ask you, does this look like, is it most consistent with autosomal dominant, autosomal recessive? We're going to do autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and Y-linked. I'm not going to ask you about any of the diseases or anything else that are autosomal dominant. So just look at the genetics of what you see here, tease it apart, and see what's going on. So here we have autosomal dominant. First thing you notice, first thing I would be doing if I looked at any of these was, is it sex specific or is it affecting both sexes equally? Here it's affecting both sexes about equally, so that's pretty strong indication it's an autosomal mutation. Dominant, well, first you begin to look at it, you notice right away that you probably will have affected individuals at every generation. You can look at it a little more closely, okay? So, for instance, an affected individual will always have at least one affected parent, right? Unaffected individuals will have unaffected parents. Autosomal recessive, you can have unaffected parents but have an affected child, okay? So if we compare, so that's really what this just sort of summarizes. Okay. So again, this so-called skipping generations, so I think this is everything we just said. Now, if you just compare that to autosomal recessive, it's just like what Mendel was looking at. So this is going to be just like Mendel's. The heterozygote will be, the two heterozygote parents who could be heterozygotes are going to be unaffected, but then they could have an affected child. That's going to occur about 50% or 25% of the time. So let's get to okay, here we go. So here's the difference. Now you have an affected individual, but you have two unaffected parents. So that piece of data right there can tell you the difference between dominant, whether it's a dominant trait or recessive trait. And here's where you get this so-called skipping of generations. So unaffected, affected, unaffected, and then affected again. And again, the highest probability comes between two related individuals. And that is for most of these mutations, especially ones affecting humans, most of them are going to be rare. So, all right, and probably an important point to put down when solving these problems and such, if it's a dominant mutation, the affected individual is almost invariably a heterozygote. Why would that be true? If the affected on these dominant mutations, okay, invariably the affected individual is a heterozygote. Why? Right. Well, if the affected child was right. Okay, so but if the affected individual is homozygous dominant, then both parents would have to have the trait. And that's probably extremely rare. Most of these traits are very rare in the population anyway. So that's why in these recessive traits, they usually are most frequent the more closely related the individuals are, because it's the higher probability that that particular mutation is in that genetic line. Okay? Otherwise, if these are fairly rare, even if this affects 1 in 10,000, if this mutation is present in 1 in 10,000 people, for these kind of mutations, that'd be very high. But two randomly picked unrelated individuals, the probability that they both have that becomes we go back and we multiply our probabilities, and you now have 10 to the 4th times 10 to the 4th, or 10 to the 8th. So you're at about 1/8 of the population of the world. And so the probabilities are very low. Now, if they're closely related, so that allele is already in that genetic line, then the probabilities go up. Okay. All right. So here's a couple of, that's just what we've been talking about. X-linked recessive. So this is the classic color blindness. All right. You're going to have this very distinct pattern of inheritance. Will it only affect one sex? No. You can have colorblind females. It's just going to be more prominent in males because they only have to have the one copy of the recessive allele. The female will have to be, for females, it's just like an autosomal gene. You have to be homozygous recessive because of the 2 X's. Okay? So it is most frequently in males, but it can occur in females. Again, it's going to tend to skip this generation because sons inherit the X chromosome from the mother. The mother's probably going to be a heterozygote and not have the condition, and so it goes from mother to son, and then from son to daughter, and then from daughters to the next generation. So you have this kind of skipping of generations. Okay? So if you have a heterozygous mother or carrier, if you want to use that term, half of the sons will be affected. The daughters will only be affected if the father's colorblind and the mother's at least a heterozygote. Okay? So let's see. X-linked recessive traits, so they're never transmitted from the father to son. So if the father is affected, if you look at the pedigree, if you have an affected father, you'll see no affected sons. Why is that? Because the Y chromosome is going. Real basic, okay? If you have an affected father, then all the daughters will be at least heterozygotes because they're getting the X from the father. A mother to be affected will have to be homozygous, and therefore, if the mother is actually colorblind, then all of the sons will be colorblind because she's homozygous recessive. Follows all the Mendelian rules. Okay? So that's what you see here. This dot indicates a obligate heterozygote. So for these individuals, it doesn't mean we've identified all the heterozygotes. It just means we know that these individuals must be a heterozygote. Okay, so here you have an affected son. All right. So the mother but the mother's not affected, but she has to be a heterozygote. Okay. So that's just like we've seen before. Any questions? Okay, so here's the pedigree for the royal families of Europe. And what they had was with the hemophilia. But what you notice also, if you look through here, one is the affected sons most of the time do not have children. But basically, by the time you get to this generation, these affected individuals here, I think what happened here is all the affected individuals, none of the affected sons had any children, and then it just disappears from the line. Okay. Later on, when we talk a little bit about genetic counseling, what you should be able to see from this, and this is kind of an example, sort of getting there. If there's a particular genetic disease that's in your family, a recessive mutation, and it persists from generation to generation because of the heterozygotes. If you could identify the heterozygotes and convince them not to reproduce, cash payoff, whatever it was, okay? But this is a theoretical question. But if none of the heterozygotes have children, then basically that recessive allele has been eliminated from your family. Right? Okay. X-linked dominant, a little bit different because it's now a dominant mutation. So in this case, an X-linked dominant will actually more likely affect a woman. It occurs at a higher frequency in women than in men. Okay. The trait here is always passed from the mother to the son. All right, just like we saw before. Same as X recessive. But now females can inherit the X-linked trait from either parent. All right? So this is where it changes a little bit. So you don't see, so if you're looking at the pedigree, and it becomes very clear that it looks like it's sex-linked because it's predominantly affecting one sex more than the other, but you see it's now predominantly affecting females and not skipping generations, and that's more likely that you're looking at an X-linked dominant mutation. All right? So if you have a father who has the dominant allele, all of the daughters will be affected. All right? So you look on the pedigree, you got an affected father, all the daughters should be affected, but none of the sons. The affected woman, as I said before, is probably a heterozygote. So she'll pass that allele and be there. Tools and the information that's available to human geneticists in trying to work out specific questions related to human genetics, disease, and such. And I think the other thing to sort of think about is we can't really answer this question yet is how will some of these things such as genetic counseling and that, how are they going to be changing in the age of genomics, and especially the fact that the cost of sequencing genomes is falling so fast. And I think it'll either augment this or completely supplant some of the things that we're doing now. But here's sort of what's available now. You probably have heard about these before using twin studies. So monozygotic twins are basically nature's cloning experiments, right? So there should be, as far as we can tell, genetically identical, whereas dizygotic twins are two separate eggs fertilized by two separate sperms. So those two twins, also called fraternal twins, are really no more separate, or they're no more genetically identical or genetically different than any siblings. Although there is a difference. It is because they're raised in a common intrauterine environment. Okay? So that's another factor that comes into play when you, like with surrogate mothers and such. It's a little complicated, but because of the mother and the intrauterine environment, the same, let's say, cells that are clones of each other don't give rise to identical individuals. But putting that caveat aside, what people look at is something called concordance, which just simply means how good of an agreement do we have? We're really looking at two factors. We're trying to argue whether this particular trait we're looking at, is it mainly driven genetically, or is it environmental? So what we're doing is the concordance, a high concordance, would be a very high correlation between the particular environment you're looking at, let's say, in the particular trait. I think it's going to be easier to just show you some examples. So high concordance just means we're going to see a high percentage. Discordance means a low percentage. So as it should make sense, if you're looking at a genetically influenced trait, what you would expect to see is a higher concordance among the monozygotic twins because they're genetically identical. Okay? But it's complicated. It could also be a common environmental factor. You can't really rule that out. That if you look at, here's some studies on asthma from twins, what you see is 65% concordance of monozygotic, but only 37% for dizygotic. Okay. So it's, again, strongly influenced by genetic factors, but there's also environmental factors. Okay. These kind of studies, I don't like. This is not the kind of research I'd be drawn to because I want to have more control over the system. But that's the limitations that you have. Yes. No, I doubt if you ever see 100%. You'll just see at sort of a high level. I think I got another example that's going to come from adoption studies, so that you can see the difference in the concordance. That it's actually got a very strong genetic component. So these are kind of relative. It's kind of squishy. It's kind of, it looks like there's a major genetic component, but there's also environmental components. And at least as a researcher, I don't know really where to go with that information. So that's why I personally just like using like Drosophila because we have so many tools. We can control the situation. The other advantage of working with Drosophila, if you're working in anybody's lab, there's very, very strict rules and guidelines for working with animals in research. All right? There are people living in houses that would not pass the standards for research animals. The standards are really quite high for research animals. And if you're working with a vertebrate, you have to have a whole protocol in place and have it reviewed by an institutional animal care committee and such. Drosophila, since it's not a vertebrate, I can do anything I want to it. Okay? I was at the University of California's genetics department out in University of Davis, UC Davis, out in California before I came out here, and it at one point was like the top genetics department in the country and had all these Drosophila geneticists. But the med school and such also has all these animals. They have primate colony and such. So they were visited regularly, like by PETA and other different groups. However, it turns out there is no Drosophila liberation group. There's nobody. We never had anybody break into the lab at night to release the flies. And, we killed 10,000 flies in an afternoon for some biochemical experiment. Nobody blinks, but it seems a little hypocritical to me. But it's kind of the fin. And if it makes a good pet, then people are concerned about it. If it's not a pet, then they're less concerned. So. Now, I step off my soapbox. Okay. All right. The other way to go is with adoption studies. So to take biologically related siblings, but then have them in different... The number of human disorders and diseases that have been mapped out and connected with specific genes is really quite large. So there's over 15,000 known human diseases, disorders, and such. If you want to just kind of get some information, look around, there as a reference, you can go to NCBI. So this is probably, if you do a Google search NCBI, you'll get the home page for it's the National Center for Biotechnology Information. This gives you links to like the PubMed database for searching the biomedical research literature. Get you into GenBank DNA database, which is actually it's freely available to anyone with an internet connection. So this is literally your tax dollar at work, maintaining that database. This is the online Mendelian inheritance in man, so it's more specifically targeted to human genetic diseases. Maybe a little more organized than this. There's something called gene cards. And if you go there, there's it's like built around sort of an old-fashioned index card. But you can pick out, there's like the disease of the week and hot diseases and find out what you want. And if you should poke around these things because just to see how much information is right there at your fingertips. On that one card, on that one page, it tells you almost everything that's known about that particular gene, the references in the literature, what the phenotype of the mutants are, if the protein's been characterized, what its biochemical reaction is. A lot of it is very commercial. There's companies who want to sell you antibodies against that protein and such. It's getting harder for me to put a bioinformatics assignment together for biotechnology lab because the data's all there. If you just look in the right place, they practically have the answers for you. So that's the other thing. Just do it, just to see how much information is right there at your fingertips. Okay. So this lets us kind of get into this area now of genetic counseling. So sort of the old-fashioned way, genetic counseling really was limited to, well, let's see, Uncle Harry had such and such a disorder, try to put these pedigrees together, make some probability predictions about the possibility that these two potential parents might have a child with a particular disorder, talk about probability of Down syndrome and such, but it was pretty crude compared to what we can do today. Personally, I don't know what the predictions are for genetic counseling as a career, but I think it's going to be a very growing field. Everybody probably heard about that 23andMe product that was, I guess, was going to be sold through drugstores, and you send in a DNA sample. But the problem with that is the average person who doesn't have the training in the genetics is going to get all this information and won't know what to do with it. So I think as more and more genetic profiling is done, somebody's got to help people understand what it really means here. In many cases, it's involving probabilities, this sort of thing. You know, you've all seen the drug commercials around dinner time, and you know they go on and on and on about why do I have to know what all these side effects? That's what the doctor's getting paid for, right? Okay, ask your doctor about this. And I also like that they find, you know, a side effect of death. Yeah. Sometimes happens. But what they never tell you in any of that is what the probability is. Is my chance 1 in 100 or 1 in 100,000 about it? So it's a lot of statistics involved in this. Okay. But just to get a few things sort of, so what this section's laid out is, can kind of walk you through what sort of genetic counseling and what technology is available sort of at the sort of pre-pregnancy state, maybe in vitro fertilization analysis, and even post-implantation. And just to kind of give you a feel for what the technology is able to do today. So the whole idea of this is, you know, genetic testing, especially if you suspect, you know, maybe you and your partner or someone in your family who each had cystic fibrosis. So there's a possibility that each of you may be carriers for this, and then there's a 1/4 chance that your child would have the disease. What's changed, first of all, is instead of just doing probabilities, you can just test the individuals directly, and you can find out for sure. Okay. The other is sort of post implantation, or so so the mother's already pregnant. You can do ultrasounds. Some of the ultrasounds are getting very high resolution. They may pick up very distinct malformations and such. That's possible. The other is fetal karyotyping. So everybody knows about this. It's ultrasound. It's amniocentesis. What you're really doing is looking at the chromosomes from the fetus, primarily looking for chromosomal aberrations like aneuploidy, such as chromosome triploid for chromosome 21 and such. Now, the caveat to that is if that comes back as a nice, clean analysis, does that mean there's no problem? No, it just means they didn't find anything, because they're really looking for very gross. We'll talk about this later on when we talk about chromosome rearrangements, but any change that we can actually see in the chromosome under the light microscope involves a very large number of genes. So it could be very, very small chromosomal aberrations that are not going to be picked up by this kind of karyotyping. Right, and it wouldn't show up. Now, what you can do with these cells too is take the DNA, and you can also do, if you suspect a specific genetic disorder, then you could also test for that. So the two widely used techniques, amniocentesis has been around for a very, very long time. So you're really taking a sample of the amniotic fluid that surrounds the fetus. The main thing here is the time factor. And so you're normally around the 15th, 18th week. You get the cells. You don't get enough cells to test them initially, so you have to kind of grow them in culture. So you're not really getting the results. You're almost 18 weeks into the pregnancy at that point. Although the risk of miscarriage is there, but it's usually fairly low. It's about 0.25, or maybe 1 in 400 procedures will induce a miscarriage. To be able to do this, a popular technique that probably close to 30 years old now is chorionic villi sampling. So the chorion cells are actually a part of the placenta, but they are of fetal origin. The main advantage here is you can do it sooner. You get a larger cell sample, so you can do basically a preliminary karyotyping, then grow them in culture and test it again. The downside is it has a slightly higher risk. The risk of miscarriage is around 1% with these two techniques. The more ideal situation, and this is probably becoming a little more widespread, because I think this was probably about 20 years old now. The whole idea was fetal cells, the story was always fetal cells don't cross the blood barrier, the blood placental barrier. and get into the mother's bloodstream. But in fact, they do. A very, very small percentage. The idea is, how do I capture, in a blood sample, how do I capture the few cells that are of fetal origin and separate them from the mother's cells? So there's a very sensitive, so here's one that's been tested for a long time because it does cross from the fetus, alpha-fetoprotein. This is the one. Elevated levels are indicative but not conclusive for neurotube defects. But we can get more specific with this now by using something called FACS, fluorescence activated cell sorting. So literally by having an antibody that specifically reacts to antigens that are unique to the fetal cells, the FACS can literally sort of shoot cells through a stream one cell at a time and sorts them out. So it is a technique for being able to isolate just literally a needle in the haystack, just a few of the fetal cells, and now you have a much less invasive technique. You can get the fetal cells, especially if you're doing DNA comparisons and such. You can use the father to help determine that these cells are in fact of fetal origin. And so this is getting better now in terms of being able to do this type of analysis without the more invasive techniques of CVS and amniocentesis. In fact, Dr. Hartman just got a new FACS. So he's actually thought about offering a course in it if there were enough students who were interested. But, okay. You can also do pre-implantation genetic screening. So let's go back to the cystic fibrosis scenario. So it turns out that both parents are heterozygotes, so there's a 25% chance the child will have the disease. I don't like those odds. Those are pretty high for me, but I don't want to adopt because my only link to immortality is to pass my genes on to the next generation. Okay, you have to have your own genes present. At least one of my former professors, that was his argument, that was our only link to immortality, is the genetic information we pass on to the next generation. So what you can do is you can do in vitro fertilization, and then this fertilized zygote, you allow it to begin to divide under laboratory conditions. Some experiments that were done probably 20, 30 years ago in mice showed that at the early stages, around the 8 to 16 cell stage, this differentiation really hasn't started yet. And any of those cells could in fact give rise to a whole individual. What you can do is remove a single cell, and with the polymerase chain reaction, because it's so sensitive, and this is very routine now, you can do a DNA analysis from the DNA you get from a single cell. And so you can screen that embryo to see if it was homozygous recessive for the cystic fibrosis gene. If so, then you go to the next embryo, because do in vitro fertilization, you always have multiple embryos. Okay, that's where you're trying to prevent a very debilitating disease. But you can also screen for other things. This has been done in many areas, screening for sex. If you want a male or female, you can screen through, use a probe for the Y chromosome, and determine whether it's male or female. But this is a way to screen pre-implantation. I think a lot of this is being done fairly routinely now. Okay. That's just what I was saying here. It sort of raises these ethical issues of where do you draw the line. As we do more genetic profiling, we do get more information on more genomes. We're going to find out which alleles of which genes contribute to certain traits, maybe particular hair colors, eye color, intelligence, athleticism. And although we're not at the point of manipulating the genes of the parents, you might look at it and go, okay, here's the optimal combination I'd like. Now I'm going to start screening these embryos till I get the one that I want. Okay? Don't know. That's just what could be done. Okay. Postnatal screening. We've talked about this before, and this has been done routinely for years. Blood tests for things like PKU on newborns. I'm still thinking that it won't be that many years before a whole extensive array of alleles of, let's say, the 5,000 genes most likely be involved in some sort of disease. That'll all be screened, and that information will be out there among newborns. Okay. This other one is this heterozygote screening. This is really a really good example of this is Tay-Sachs. In certain populations, the allele of that debilitating disorder is present at a fairly high frequency, and there's a lot of intermarriage in that within this group of individuals. So the probability that the parents are heterozygotes is much higher. If you think that's there, we can basically just do the genetic screening. We don't even have to do probability. You can actually take DNA sample, screen for the alleles directly. And the prospective parents will know ahead of time what the probability of having this particular disease. Pre-symptomatic. This one gets a little more controversial. Not controversial, but really starts to get into social and ethical issues. When you're dealing with these late onset diseases, like Huntington's, and it's a dominant mutation. So if there is a history of Huntington's in the family of either parent, then there's a concern that one of the parents might have that dominant allele and therefore might transmit that to their child. So let's say you're in your mid-20s, although that's too young to have children. I was a father when I was like 35, and that was a little too young. But anyway, so back to serious. So you have the parents get checked. The problem is if it's a recessive mutation, cystic fibrosis, one of these other ones, you're not impacting the life of the parents. You're just saying, look, there's a certain risk that your child might have this disease. These dominant mutations, these late onset dominant mutations, you're literally reading the future. So the question would be, you're 25 years old and there's a history of Huntington's in your family. You want to know whether you have that allele when you're 25, and that somewhere there's a ticking time bomb, and somewhere around age 40, you're going to start developing this highly debilitating and ultimately fatal nervous disorder. Or would you rather just wait and see? Or would you rather wait? Would you like to know whether you got a clean bill of health, whether you don't have the allele at all? And different individuals will have different preferences. Okay, but it raises some strong ethical and psychological issues and such to be dealt with. The information's out there. Okay. The other is just genetic discrimination, and there are cases of this already have been done. If we start doing this wide-scale genetic profiling on newborn children, and everybody knows that once information gets into a computer database, it is secure. No one can get in there. No one can ever get that information, right? Raise your hand if you believe that, and you shop at Home Depot. All right. So the information is going to be out there. The question is, how do we as a society deal with that information? So regardless of which side of the political fence you're on, I think a big part of the Affordable Care Act was not allowing insurance companies to pick and choose their clients based on pre-existing conditions. Okay? To me, that was always a little bit like trying to play blackjack out in Vegas and the dealer gets to see my cards. It's just not fair that way. Okay. Getting off subject a little bit, but it's something I find really interesting, is evolutionarily, could small mutations really set the stage for an evolutionary pathway? So just as an example, we've talked about this before. Humans and chimpanzees share about 95% homology of the DNA. So the interesting thing is, what about the other 5%? What's different there? Well, there's a gene called FOXP2. This gene's been implicated in human speech. There's several speech pathologies that are known to be associated with mutations in this gene. And this is one of the genes that's quite different between primates and humans. Yes?
Is it possible to insert the gene into primates?
Oh, they have it. It's just a different version. But that would just be the initiating step. There's also been other events. The key here is, did this one event sort of set the stage for a divergence? In other words, the limb in that tree of the common ancestor in humans diverged this way, and chimps diverged this way. Okay. The idea is, could this simple mutation that maybe only occurred as little as 200,000 years ago now set the evolutionary path for speech development, possibly leading to written records, and really the differences that distinguish humans from the primates? Just something to think about.